Welcome to our YouTube channel Biology Board. Today we are study and discuss about process of transcription in prokaryote. Now, according to NCERT, the process of copying genetic information in form one strand of DNA into RNA is known as transcription. Now, according to this, transcription is a process in which ribonucleic acid RNA is synthesized from DNA. The word gene refers to the functional unit of the DNA that can be transcribed. Thus, the genetic information stored in DNA is expressed throughout RNA. For this purpose, one of the two strands of DNA serve as a template. Now, what is template? The strand which is used for synthesizing RNA is polarity in 3' to 5' direction is called template strand or non-coding strand or sense strand and produce a working copies of RNA molecules. The other DNA strand which does not participate in transcription is referred to as a coding strand or antisense strand. Now next is before transcription can take place, the DNA double helix must unwind near the gene that is getting transcribed. The region of opened up DNA is called the transcription bubble. Here we can see this is a DNA. Now unbind DNA helix called transcription bubble. This is coding strand, this is template strand, and this is newly formed RNA strand. Transcription uses one of the two exposed DNA strand as a template. This strand is called template strand. The RNA product is complementary to the template strand and is almost identical to the other DNA strand called the non-template or coding strand. Now, the site on the DNA from which the first RNA nucleotide is transcribed is called the +1 site or the initiation site. This is the place where first nucleotide is transcribed. It is called initiation site or +1 site. Nucleotides that come before the initiation site are given negative numbers and said to be upstream. Now this is initiation site. Upstream come from before initiation site. So this is completely upstream having negative number like -1, -2, -3, -4. Nucleotides that come after the initiation site are marked with positive numbers and said to be downstream. This is downstream come after initiation site and they have a positive numbers like +1, +2, +3, etc. This is whole gene having promoter and terminator site and transcription unit also called as a structural genes. Next, a single enzyme DNA dependent RNA polymerase. Now, enzyme responsible for transcription is known as RNA polymerase with help of DNA. So, it is also called as DNA dependent RNA polymerase with 5 polypeptide subunits, 2 alpha, 1 beta and 1 beta dash and 1 sigma factor. Now, what is sigma factor? Sigma factor helps RNA polymerase to bind with promoter and initiation of transcription. The enzyme with sigma factor is known as a hollow enzyme, and without sigma factor it referred as core enzyme. This is core enzyme also called as RNA polymerase with sigma factor, now completely called as a hollow enzyme. Transcription involves 3 different stages: initiation, elongation and termination. Now initiation, the binding of the enzyme RNA polymerase to DNA is the prerequisite for transcription to start. Now what is prerequisite? Prerequisite means things that is required. Here RNA is essential for transcription, so RNA is the prerequisite for the transcription to start. The specific region on the DNA where the enzyme bind is known as a promoter region. There are two base sequences on the coding DNA strand, which the sigma factor of RNA polymerase can recognize for initiation of transcription. This is two base sequence which is form promoter region and where the RNA polymerase can recognize for initiation of transcription. Next, first sequence is Tata box sequence, second is -35 base pair sequence. Now, Tata box sequence, this is the first recognizing site in the promoter region of DNA. It contains a base sequences TA, TA, AA, which is 5' to 3' direction. It's recognized by one of the general transcription factor, allowing other transcription factor and eventually RNA polymerase to bind. Second is -35 sequence. This is the second recognizing site in the promoter region of DNA. It contains a base sequence TT, GA, CA, which is located about 35 bases upstream. This is a upstream. Hence -35 away on the left side from the site of transcription start. Now one is Tata box sequence and other -35 sequence. Both combined and form promoter region. Here you can see both combined and form promoter region where RNA polymerase with sigma factor bind. And both RNA polymerase with sigma factor help to unwind DNA double helix. Now sigma subunit dissociate and RNA synthesis is initiated. Next is here in this figure you can see this is the promoter region, this is upstream and this is downstream. Now RNA polymerase with sigma factor bind in promoter site. Next double helix of DNA structure unwind. After unwinding of DNA double helix structure, sigma factor dissociate and RNA synthesis is initiated. Now elongation. After transcription initiation, the sigma factor is released from transcriptional complex to leave the core enzyme, also called as RNA polymerase, which continues elongation of the RNA transcript. The core enzyme contains a catalytic site for polymerization, probably within the beta subunit. The first nucleotide is the RNA transcript is usually 3PG or 3PA. The RNA polymerase then synthesizes RNA in 5' to 3' direction using the 4 ribonucleoside 5' triphosphate ATP, CTP, GTP, UTP as precursors. It is, however, identical to that of coding strand except that RNA contains U in place of T in DNA. This is template strand, this is coding strand, this is newly formed RNA, and this is RNA polymerase enzyme. Now, next we see the whole concept of elongation. The complex of RNA polymerase, DNA template, and new RNA transcript is called ternary complex. This is whole called ternary complex, and the region of unbound DNA that is undergoing transcription is called transcription bubble. Now, during elongation, RNA polymerase walk along one strand of DNA, known as the template strand, in the 3' to 5' direction. For each nucleotide in the template, RNA polymerase add a matching complementary RNA nucleotide. This is template strand, and RNA polymerase add a complementary nucleotide in sequence of 5' to 3' direction. 3' end of the RNA strand. Now the DNA is unwound ahead of the transcription bubble, and after the transcription complex has passed, the DNA rewind. Now the strand which is used for synthesized RNA has a polarity in 3' to 5' direction called template strand. Here we can see thymine is replaced by uracil during RNA formation. The sequence of DNA which form mRNA is known as structural gene, and this process known as elongation. Now termination. The process of transcription stop by termination signals. Two types of termination are identified: row dependent termination and row independent termination. First one is row dependent termination. The RNA contains a binding site for protein called row factor. This is row factor and this is binding site contain RNA. Row factor bind to his sequence and start climbing up the transcription towards RNA polymerase. Row pulls the RNA transcript and the template DNA strand apart, releasing the RNA molecule and ending transcription. Another sequence found later in the DNA called the transcription stop point causes RNA polymerase to push and thus help to row catch up. In this figure, we can see row protein bind with row binding site in RNA strand. They climb up and dissociate the RNA strand to the RNA polymerase. Next is row independent termination. Depends on specific sequence in the DNA template strand. As the RNA polymerase approaches the end of the gene being transcribed, it hits a region rich in C and G nucleotide. RNA transcribed from this region fold back on itself and the complementary C and G nucleotide bind together. Here we can see C and G strand, complementary strand bind together. The result is a strand hairpin that causes the polymerase to stop. The hairpin is followed by stretch of U nucleotide in the RNA which match up with A nucleotide in the template DNA. The complementary UA region of the RNA transcript form only a weak interaction with the template DNA. This, coupled with the stalled polymerase, produces enough instability for the enzyme to fall off and liberate the new RNA transcript. This is the whole concept. Thank you for watching. If you like this video, please like, share and subscribe. Thank you so much.